Ribosomes are one of the most important organelles. You have tons. They are essentially a glob of RNA and protein, two groups that stick together that make more proteins. Many are attached to the rough ER or just lie scattered out in the cytoplasm. They are the site or the place where protein is made, protein synthesis. Ribosomes in the endoplasmic reticulum make proteins to be sent out or to be put into the plasma membrane. Free ribosomes make proteins that the cell uses itself. So particular enzymes the cell is going to be using inside of the cell. They kind of look like this. Two little globed ribosomes. They're really made up of what's called RNA, the scaffolding that kind of attach together. Special RNA called rRNA or ribosomal RNA. But they're two subunits that join together and they read mRNA or messenger RNA and allow a cell to know how to attach amino acids. So we just learned that proteins are a bunch of amino acids linked together. But when the ribosome is making the amino acids, someone has to tell them what order to put that amino acid in. And so it is the rRNA, the ribosome, that reads that mRNA and knows what order to put the amino acid in. We name ribosomes based on their sedimentation rate, or what we call a Svedberg unit. They come in different names like 80S, 70S, 50S, 30S. We have a very particular kind of ribosomes versus bacteria ribosomes, which are different. But interestingly enough, bacteria ribosomes are the same ribosomes we find inside of our mitochondria because they are essentially just little bacteria inside of our cells. And so we have to be very careful because some medications that we take to attack bacteria attack the ribosomes, and if taken too long can begin to attack our own mitochondria. And so when I say different units, there's one unit and another unit that attach together, and the mRNA, the template for how to build a protein, feeds through that. And the ribosome reads it one at a time and knows what order to put this protein. But they're very, very tiny, 25 nanometers. That's impossibly small. If we have our nucleus in the middle of a cell, branching out from the nucleus and physically attached to the nucleus itself is what's called the rough ER, or the rough endoplasmic reticulum. It's an interconnected system of canals, or the fancy science word for that is cisternae. It just means openings. They're continuous with the nuclear membrane itself, so they're attached to it. And there are two forms, the rough that's covered in ribosomes and the smooth ER that does not have ribosomes. So the rough ER actually branches off from the membrane of the nucleus and is filled with these ribosomes. That's because the rough ER is going to fold proteins into its particular shape, into the primary, secondary, tertiary shape. And so if you have the little protein building factories here, it can go right to the ribosome or right to the ER and they can be folded. But let's not forget about the smooth ER. It doesn't have ribosomes. So if you're trying to remember function, right, you're going to have to be able to help yourself out on the test. The smooth ER doesn't have ribosomes. So if you remember that ribosomes build proteins, then obviously the smooth ER probably isn't going to be doing anything with proteins, or it would be covered in ribosomes. So the rough ER is, they say, studded or covered with ribosomes, and it manufactures all of those proteins that are going to be secreted, proteins that are going to leave the cell. They're responsible for the synthesis of integral membrane proteins and phospholipids for cell membranes. Are you okay so far? No one's saying anything in the chat, so I assume you're keeping up. So we're about to get 2 chapters worth of information in one little piece here. So the information for making a protein is inside of the nucleus. That's our DNA, our genes. We cannot let DNA leave the cell. It would break down. So what we do is we make copies of genes called RNA. Specifically, we send out a very special type of RNA called messenger RNA. That's just a copy of a gene that leaves the nucleus and then can be read by the ribosome, and that's how we make a protein. Then we recycle the RNA fragments and do it all over again every time we need to make that protein. You can't send your DNA out. That's your precious only copy. So you make versions of it, little copies of it to send out, which are read by ribosomes to make proteins. That's gene expression right there. That's a whole branch of science, going from DNA to RNA to protein to everything that you are. So DNA is the information. We make an mRNA strand. The mRNA can leave the nucleus, be read by the ribosome, and allow us to make that polypeptide or make that protein. The portion where we make an mRNA copy out of DNA is called transcription. The part where we make proteins from that information is called translation. So we transcribe or write the mRNA and then translate it into a protein. Smooth ER is also branched off from the nucleus, but it doesn't have any ribosomes. So it's not necessarily going to be doing anything with proteins as its main job. It's a set of tubes arranged in a network, but it allows for various processes, and you're going to need to know some major ones here. In the liver, the smooth ER is responsible for lipid and cholesterol metabolism. Really, when you think about smooth ER, think fats. So when you think rough ER, think protein manufacture. When you think smooth ER, think fats. It's not its only job, but that will help you a little bit if you think fats. So now that you have smooth ER equals fat in your head, in the liver, that's where we're going to handle fat, lipid, and cholesterol metabolism. I said that if we have too much sugar, we can store it as glycogen. So the smooth ER is going to help us break down glycogen. And along with the kidneys, the liver, the smooth ER and the liver and the kidneys help detoxify our systems, help break down drugs and medicines and various chemicals. Specifically in the testes, the smooth ER allows for the synthesis of androgens and hormones. But testosterone is a lipid, and so it's going to diffuse right on out of that cell, right through that plasma membrane. In the intestinal cells, smooth ER allows for the absorption, creation, and transport of fats. And here are two big ones. Your muscles need calcium. So the smooth ER in skeletal and cardiac muscle is the place for storage and releasing of calcium ions. Now the smooth ER does a lot of other stuff too. but you should definitely know these four. Liver, lipid metabolism, testes, steroid hormones like testosterone, intestines, transport and absorb fats, skeletal and cardiac muscle, store and release calcium. Very similar to the smooth ER, a system of channels called the Golgi apparatus. It actually is broken off of the ER. It's not fused with it, but it is its own separate thing, a system of faces. Those proteins and fats that we just made move through these faces one at a time. And every time it hits a face, it's packaged and a special sugar is added. And that's how it knows how to leave or where to go. So the Golgi apparatus is kind of like the packaging facility. It functions in modifying, concentrating and packaging proteins and fats. Transport vessels from the rough ER fuse with the Golgi apparatus and then can be released. The Golgi is also a place where we can break off little pieces here and make lysosomes or digestive vesicles, things that are going to break down fats and other materials. So proteins pass through that Golgi apparatus, each of those faces, and then the secretory vesicles can then move to other parts of the cell or leave the cell. Here's an actual image of the Golgi. Isn't that absolutely amazing? And so you can see it's just a bunch of different faces. And if here's the nucleus, here's the ER, as the stuff we make from the smooth ER and the rough ER makes its way through the Golgi, special sugars are added and tells it where to go. So here's a good picture to show you that sometimes the pieces that break off from the Golgi, you can't just send this whole vesicle out. It can actually fuse with the outer plasma membrane and then dump its contents out. So if you made a hormone here or an antibody or some other protein that we just dump out, now it can leave and enter the bloodstream and be sent elsewhere throughout the body. Oppositely, sometimes we have to bring things in the cell and break them down. And so little pieces of the Golgi is broken off and we can form what's called lysosomes. Zome just means body. Lyso means to break or cut down. And so lysosome is breaking down. It's a little spherical circle filled with enzymes that can help us break down contents that we don't need, or invading pathogens like bacteria, viruses, and toxins. They help us recycle things too. So lysosomes are going to help us break down non-functioning organelles, help us break down glycogen, help us break down bone when we need access to calcium, and help us release thyroid hormone, which controls amongst other stuff, bone breaking down or building up. It can also break down irrelevant tissue. And here's what I was talking about. It can help us break down bone and release calcium, which that's calcium 2+, the ion. And so we can find these lysosomes in our white blood cells, the immune cells that are going to go out and attach to pathogens and break them down, as well as melanocytes, the melanin-containing cells inside of our skin. Interestingly enough, if you get a tattoo, that pigment is consumed by the lysosomes inside of these melanocytes, but they can't break it down fully, and so it sticks there. Over time, it diffuses as new skin cells are formed, And so over time, you know, the barrier, the outer line of your tattoos, excuse me, kind of like lose their sharpness, but we can't truly break down that pigment all the way. And so it's consumed by these lysosomes and these melanocytes, and it sticks there, it's stuck there. That's kind of cool, right? Maybe, I don't know. And so it looks like a little sci-fi creature here. And so if we were talking about a little bacteria here, we can absorb it, seal it off, and then we can send in a lysosome filled with a digestive enzyme. It's going to release that enzyme and then break all of that down. And that's essentially what's happening when you have a you know, invader bacteria. Your body tries to break it down faster than the bacteria can make you sick. Now, interestingly enough, and maybe you know this, maybe you didn't. When I learned it, you know, a decade ago, I thought it was wild, but you know me by now. I think everything is wild and interesting when it comes to the human body. But did you know that when we are infected by a bacteria, oftentimes it's not the bacteria itself that is initially making us sick. What gives us all of those terrible symptoms, like our nose running, the fever, the headache, you know, just that general malaise, and you know, we hope that our body, if it's one of the types of bacteria our body can fight off, you know, we survive it. But what is causing us to get so sick are the toxins that are released when we break down the bacteria, when it's dying and dead. But it's what you got to do. Anyway, I thought that was wild. Because you always think you get sick because your body is, you know, busy fighting it off. And it is kind of, that's true. But the main part of sickness sometimes is just the toxin released when a bacteria dies. That's why sometimes it's a sign that you're getting better, if it is a disease that you can get better from.